Assalamu alaikum. Today we are going to talk about the heredity material, the chromosomes, DNA and genes. In this lecture we are going to cover certain goals. Goals of this lecture are structure of the chromosomes, structure and functions of genes, structure and functions of DNA and the heredity information flow, transcription and translation. We start from what is a heredity information flow. Heredity, something that is transferred from one generation to the next generation, that is from parents to the offsprings. Reproducing itself is a property of life and is the essential property of life. This is the characteristic of all the living organisms, that they reproduce themselves and they transfer their characteristics from one generation to the next generation, that is from parents to the offsprings. Heredity information flow in living organisms is carried out by the structures, or we can say structural units, called the genes. Genes are present in the form of DNA molecules, and the DNA molecules are organized in the structures called chromosomes or the chromatin material. We describe it as the chromosomal theory of inheritance. That is, there is a theory that is proved by various scientists by so many experiments that chromosomes are ultimately responsible for transferring the characteristics of the parent to the offspring. By conducting many experiments, various scientists at different locations in the world proved that without the DNA, genes, chromosomes, the heredity information could not be transferred. So the DNA molecule which have genes is responsible for the heredity information flow. Here we look at a diagram which show that, as we know, that all the cells do have a nucleus if they are eukaryotic cells. If they are prokaryotic cells, then they do not have a nucleus, that is a region separated by a nuclear membrane from the cytoplasm, but they do have a small region inside the center of the cell, which is called the nucleoid region. The chromatin material is present inside the nucleus or the nucleoid region. As this diagram shows to you, that chromatin material is present in just like entangled threads inside the nucleus. If we try to elongate that chromatin material, then you can see, if you go towards the right side of the diagram, you will see that this will be just opened like a thread, and we will see more complicated threads which are wound around different sets of proteins. If we open it even more, then we will see that there are small sets of proteins. These proteins are called histones. These proteins are the set of those proteins around which DNA molecule is wrapped. Around each histone set, as you can see in the diagram, there are the DNA fiber is wrapped around two times. Then if we open it further and separate it from the proteins, then we can see the DNA helix separately from the proteins. If we look, this phase or this structure is actually found when the cell is not dividing. That is, these are not condensed. But when the cell is dividing, this chromatin material is present in a condensed form. You can see on the extreme right in the form of the structures called chromosomes, which are actually highly condensed form of these molecules. We call them chromosomes. You can see the various parts of a chromosome. Inside the center, the constricted region is called the centromere, around which on the sides are the arms, and the very corner of the arms are called the telomeres. So this is about a generalized rough idea of that how chromatin material is present inside the chromosome, inside the nucleus in the non-dividing phase and in the dividing phase. So chromatin is the genetic material. Chromatin material, when we call it chromatin, it is actually mainly in the non-dividing phase of the cell. When we call it a chromosome, when this is present in the dividing phase of the cell. As we looked in the previous diagram, chromatin material actually in non-dividing phase is so thin, so sporadic, so thin that it is not visible under the light microscope. When the cell comes in the phase of division, then what happens that these chromosomes, these DNA molecules, start condensing in the prophase, the first phase of the mitosis, the cell or the nuclear division. These chromosomes condense, and then we must say in more correct terms that the chromatin material condense, and they make the condensed structures called chromosomes. So a chromatin material is a term for more thinner material which is not very visible, and the chromosome is the more right term for the chromatin material in the dividing phase when it is highly condensed and it is clearly visible under a light microscope if we stain them. There are certain specific characteristics of the genetic material which actually make it a genetic material, because you know that genetic material means that this particular molecule should be able to transmit the heredity information from the parents to the offspring, to the next generation. It should have certain important functions. Number one is that it replicates itself. Number one, that the genetic material should replicate itself. Replica means copy. It should be able to copy itself. If it is able to copy itself, then it could go to the next generation when the cell divides. The second property is this, that it should regulate the growth and development of the organism. This particular molecule should have the capability to regulate or control the growth and development of the organism in which it is present. For example, if we look at the human beings, we have so many characteristics. We have our eye colors, we have our hair colors, inside our body we make so many enzymes. Our skin is made up of cells, proteins. Our hair are made up of different types of proteins. Every characteristic should be made, and all of these things are regulated by the genetic material. The third property is this, that it allows the organism to adapt to the environmental changes. Now we know that environment is always changing. Environment is like temperature around us is changing always with the seasons. Wind blowing around us is always changing. Sometimes it is too much of wind. If sometimes there is a breeze, low one. Sometimes if it is it may be very hot. Sometimes it may be very cold. And even there are certain environmental disasters which can change the environment altogether. The genetic material should have the property that it can change, adapt, or we can say mutate itself to give the living organism a room for adapting or dealing with or coping with the changes in the environment. So there are three properties which makes a material, a molecule, a hereditary molecule. Number one, it replicates itself. Number two, it regulates the growth and development of the organism. And number three, that it allows to adapt. It allows the organism to adapt to environmental changes in the situation. Now the chromosomes, DNA and the genes. Chromosomes consist of DNA molecules and proteins. When we call a genetic material or a DNA molecule a chromosome, when this molecule is wrapped around, or we can say wound around, different types of proteins. So chromosome is a complex of DNA wrapped around different types of proteins, DNA plus proteins. In chromosomes, DNA is wrapped around proteins. Few of these proteins are called histones. These are a specific type of proteins, and they are named as histones. As we have seen in the previous diagram, that around which there are two full circles of DNA which are wrapped. But there are certain other proteins which are also associated with the DNA molecules in the chromosome, but these are not histones. We can call them non-histone proteins. So we divide the proteins associated with DNA into histones, and the non-histone proteins. So we are talking about the chromosomes, the genetic material, DNA. What is DNA? DNA is deoxyribonucleic acid. DNA stands for deoxyribonucleic acid. DNA is a large molecule. We call it a macromolecule, and this is present in all types of organisms except for a few in which the genetic material is RNA. In most of the organisms, DNA is the genetic material. DNA is different as it is present structurally in different types of cells. We know that there are cells which are called the prokaryotic cells, which don't have a true nucleus. There are other cells which are called eukaryotic cells, which includes the animals and plants. These cells have a true nucleus. In the prokaryotic cells, DNA exists as a circular molecule, though this is a chromosome, because this is associated with the proteins, different types of proteins. These proteins are sometimes different from the proteins present in the eukaryotic chromosome, but some proteins are similar to that of eukaryotic chromosomes. In eukaryotic chromosomes, their DNA molecule, their chromosome, is actually a linear DNA molecule. In prokaryotes, it was circular DNA molecule. In eukaryotes, it is a long linear DNA molecule in the form of a chromosome that is associated with different types of proteins, histone and non-histone proteins. The prokaryotes also have another form of DNA present outside their nucleoid region, inside their cytoplasm, which are small circular DNA molecules. These are called plasmids. The bacterial chromosome or the prokaryotic chromosomes, as we know that bacteria are the prokaryotes, the prokaryotic chromosomes, they have the genes for the regulation of growth, for control, for development and everything. The plasmids, which are we can call them extranucleoid DNA, circular DNA molecules, don't have essential genes, but these have certain genes for resistance to, for example, antibiotics, resistance to salinity, or resistant to extreme environmental conditions. So the plasmids give an extra advantage to the prokaryotes, particularly the bacteria, to cope with the extreme environments. In eukaryotes, as we talked about, that DNA is a linear molecule. It's a straight linear molecule. There are two more organelles which have their DNA molecule of their own. These are the mitochondria and chloroplast. Mitochondria also have a circular DNA molecule of its own, and the chloroplasts, which are we know that present in the plant cells, these conduct the photosynthesis. These also have their own DNA molecule. That is why we assume that mitochondria and chloroplast, they may be originated from some bacteria because they matches in their DNA material and in their division of the organelle with the binary fission in the bacteria. So DNA molecule in prokaryotes is a circular molecule. It may be in the form of a chromosome or a plasmid. In eukaryotes, DNA molecule is linear and is present in the form of a linearly arranged chromosome. And in the mitochondria and chloroplast, DNA is a circular molecule present inside their matrix. Next is gene. What is a gene? Gene is a length of DNA that codes for a peptide or a protein. So, gene is a part of DNA or a piece of DNA that codes for a peptide or a protein. Gene is nothing but a piece of DNA, a length of DNA. So, the chromosomes are the chromatin material is the genetic material, which is during the mitosis or division phase highly condensed, and we call them chromosomes. Organisms have different number of chromosomes. For example, humans have 46 chromosomes present inside each cell, the nucleus of each cell. These 46 chromosomes are present in 23 pairs. Onion have 16 chromosomes, which are present in 8 pairs, while Drosophila, the fruit fly, have 4 chromosomes present in 2 pairs. There are different types of organisms which have different number of chromosomes. Chromosomes also somehow determines the complexity of life. Mostly, more complex the organism, more number of chromosomes they do have. Now we talk about the chemical composition of chromosomes. Chemically, it is found by different types of biochemistry experiments and by X-ray diffraction studies that chemically DNA consists of 3 major ingredients: a sugar, a phosphate group, and a nitrogenous base. DNA is a molecule that is about 2 nanometers thick. This is running continuously, means without a break, throughout the chromosome. DNA is acidic in nature. The biochemical analysis shows that DNA in its nature is acidic. So, you know that DNA is present in huge quantities inside the nucleus, and it is packed. If we talk about DNA in a single cell, all of its chromosomes, and we simply elongate them and straighten them, these will be about 6 feet long. You can guess 6 feet, about more than the height of an average person, and this is packed in a microscopic cell and even inside its nucleus. How this much long DNA molecule is packed in a microscopic nucleus, very small one, which we cannot see with the naked eye? How is this possible? The answer is coiling and the supercoiling. We again have to look on a diagram. Look at the diagram which is in front of you right now. We know that chromosomes are present in pairs. If you look at the bottom right, we can see a homologous pair of chromosomes, the metaphase chromosomes, which are present in the metaphase of the mitosis, cell division. If we simply uncoil a chromosome and spread it in a linear fashion, then we will see that this is a highly complex molecule, wound, just we can say it's coiled. If we open these coils more, we can see in the yellow color, if you can see, the flower-like structures. These are called the solenoids. These are the lengths of DNA wound around or wrapped around the histone proteins. If we even open them more, if we open the solenoids as well, then we can see the bead-like structures. These beads, these individual beads, are actually lengths of DNA wound around or wrapped around a set of histone molecules. We call it a nucleosome. One nucleosome is actually a set of histone molecules and 2 wraps of DNA molecule around it. This DNA, which is wrapped around in 2 circles around the histone molecule, is about 200 base pairs long. Then you can see that these beads are attached to each other. This part of DNA, which is attaching them with each other, is called the linker DNA. That is, it is linking nucleosomes with each other. This linker DNA is roughly about 80 base pairs long. Then if we open this, if we still open the nucleosome and separate the DNA from the proteins or unwrap the DNA from the nucleosomes, then we can see that DNA is a very long helical structures, 2 strands going side by side together in a helical fashion. This part of DNA actually, which is now without the proteins, just 2 DNA strands going side by side together, makes the genes. It means that these are only the DNA molecules with no association proteins. In this form, these are capable of making the RNA molecule and then the proteins. We have a closer look in this next diagram on the nucleosomes. You can see 3 nucleosomes here. If we start from the right, you can see one nucleosome, a central core of proteins. We can call it a protein core made up of histone molecules. Then 2 wraps of DNA, then the linker DNA, then 2 wraps again around the next histone set, and then there is a third, and so on. If we look this structure under the electron microscope, because we know that electron microscope is a microscope which can show us extremely small structure which are not visible under the light microscope even. If we look at the chromatin material during a non-dividing phase, we can see a bead-like structure. These beads, as we can see, just जिस तरह तस्बीह के दाने होते हैं, इस तरह का स्ट्रक्चर हमें नजर आता है इलेक्ट्रॉन माइक्रोस्कोप में. This bead-like structure is actually the straightened DNA, which have nucleosomes and the linker DNA, then nucleosome and the linker DNA, and so on. So this is how this much long DNA molecule, which is extremely long, longer than the height of the a person, is coiled or is packed inside a very, very small nucleus, which is microscopic, which is not visible to us, visible to our eye. We can say that some scientist says that if we can straighten the whole DNA present in a human body and straighten it and make a thread, this thread will reach from the earth to the moon. This is that long. But you can see that how nature packed it into a very small microscopic nucleus and control the whole characteristics of the organisms using this genetic material. Now we talk about the chemical composition of DNA. DNA is a complex large molecule. It's a macromolecule. It consists of the subunits. Subunits are called the nucleotides. Each nucleotide consists of 3 major molecules. One is a sugar, that is a ribose sugar, a 5-carbon sugar, and that is a deoxyribose. The other one is a phosphate group, and the third one is a nitrogenous base. What is DNA actually? The nucleotides, they join together. That is, one nucleotide is joined to the next and next to the next, and so on. These nucleotides makes the long chains of the DNA molecule itself. And there are 2 chains running side by side, and these 2 chains are bound together. They are different parts. We'll look next. They bind together to make a helical structure. They are wound around each other in a helix. We call it a double helix because it have 2 strands helically winding around each other. If you look at the diagram, it shows you a deoxyribose sugar, a 5-carbon sugar. Its 5 corners are showing its 5 carbons. Then you can see a phosphate group on its left and a nitrogenous base, which is a 6-carbon one, on its right. This is the structure of a single nucleotide. But nucleotides even are of different natures. Nucleotides are categorized into 2 groups according to their differences in their structures. There are 2 called purines, and there are 2 others called pyrimidines. We have a look on their structure. Purines are 2. One is adenine and the other is guanine. The pyrimidines are thymine and cytosine. There is a slight difference in their structure. The purines, as you look at in the diagram, the purines are double ring structure. They have two rings. Their structure is like this, that there are two rings joined together. But in the pyrimidines, as you can see in the diagram, the cytosine and thymine, they are single ring structures. According to these four bases, we call them nitrogenous bases because you can see there are a lot many nitrogen molecules are present inside these structures. According to these nitrogenous bases, we categorize the nucleotides into four major categories. According to one, with which adenine is attached, adenosine, guanosine, cytosine, and a thymine base. So bases are adenine, guanine, cytosine, and thymine. Now we have a look on a DNA molecule more closely, how these nucleotides are arranged in front of each other. If we look at the diagram, we can see that a thymine is pairing with an adenine base. In DNA, actually, thymine always pairs with an adenine, and a guanine always pairs with a cytosine. So we can find out the GC pairs, that is guanine cytosine pairs, and AT pairs, the adenine and the thymine pairs. Now we know that the DNA is the main genetic material. It produces the RNA, the ribonucleic acid. There is ribonucleic acid is also similar in its single-stranded structure to the DNA and is always complementary to the DNA. But the difference is this, that RNA don't have a thymine. In place of thymine, the RNA molecule have another base called uracil. So if this is an RNA molecule, it will have uracil in place of the thymine, and the uracil will pair with the adenine rather than thymine, and the RNA is single-stranded. RNA don't have these two strands running together. The DNA molecule, as you can see, that there are two parallel chains. These chains are attached with each other by hydrogen bonding. If we look at in this next diagram, the guanine and cytosine bases are joined together, joined to each other by hydrogen bonding. And guanine and cytosine, the GC pair, is attached to each other at three places by three hydrogen bonds, while an AT pair, adenosine to thymine pair, is attached to each other by two hydrogen bonds. So hydrogen bonding is the force that keep these two winding strands of the DNA together. Now we come on the mechanism of gene action, how the genes they act, how they perform their functions. What are the functions of the genes basically? Replicate itself, we call it replication, making its own copies. It occurs during the cell cycle, the synthesis phase of cell cycle, when before division, before cell division, because we know that after mitosis, the cell, one cell, the parent cell, will be divided into two daughter cells, and the daughter cells should have an equal quantity of DNA as it was present in their parents. So before entering into the division phase, the DNA, the genetic material, have to replicate themselves, make their own copies which have same structure and quantity. So one function is replication. The other function is transferring the information or encoding the information present in them in the form of proteins. And this function of the DNA, it performs it by two processes, going one by one. One is called the transcription. Transcription is actually making an RNA molecule out of a DNA molecule. That is, DNA molecule act as a template. With the help of that, a complementary strand of RNA is produced. Then this RNA in the eukaryotes have to go out of the nucleus and encode for a protein. Protein may make the hairs, proteins may make the our nails, proteins may make any of the cellular structure, maybe a membrane protein, maybe an enzyme or anything. This process from RNA to protein is called the translation. So this is another function of DNA, that is transcription, making an RNA molecule, and translation, making a protein from an RNA molecule. The replication is the doubling of DNA. Replication occurs in a specific time when the cell have to divide. But the transcription and translation, these are the ongoing processes all the time occurring in the cell. Whenever cell need to make an enzyme, cell need to make any of its structures, a receptor, or anything, any structural part, it have to go by the process of transcription and translation. Then there is another related term called genetic code or codon. 3 nucleotides in the DNA codes for an amino acid. As we know that proteins consist of amino acids. Amino acids are the basic units of proteins. Amino acids join together to make protein. And 3 nucleotides combined together codes for an amino acid. We call this genetic code or codon. So a codon is a triplet code that codes that instruct the production of an or we can say attachment of an amino acid to a peptide chain. Then transcription. If just we have a brief look on a diagram, this describes that transcription is from DNA to an RNA molecule. This particular RNA molecule is called a messenger RNA. So DNA to messenger RNA is called transcription. Then messenger RNA to protein is called translation. That is, the language of DNA is translated by the messenger RNA into the protein. We call it central dogma of molecular biology or molecular genetics. Central dogma means that there is in any field of study, we have a central theory, we have a central idea around which all of that science revolves. This is the central dogma of molecular biology or genetics, that DNA codes for RNA, transcription, RNA codes for protein, translation. Now we talk about the process of transcription. Transcription is the coding of DNA into a messenger RNA. Transcription is a long process which is supported or helped out by the enzyme called RNA polymerase. As we know, polymerase is the actually addition of or making a polymer. Polymer is a long molecule. Now we simply talk about the process of transcription, that how transcription occurs. As we know that there are specific sets or we can say lengths of DNA on a chromosome which codes for a specific protein. We call them genes. The RNA polymerase have actually to ultimately create a gene. It does it by starting or doing we can say transcribing the DNA in form of a messenger RNA. How it happens? The RNA polymerase identifies a specific region on the DNA that is present before the actual structural gene. We call that region a promoter region. RNA polymerase comes and bind to the promoter region. Actually, it's a very large enzyme, and it attaches to the promoter region, it covers the promoter region, and in part the structural gene, some initial part of the structural gene. When RNA polymerase attaches to the promoter, and promoter is, we can technically called upstream the gene, the structural gene. Upstream means that it is present towards the 5' end of the DNA. When RNA polymerase identifies the promoter region and attaches to it, it starts unwinding the double helix. Because we know that RNA is a single-stranded molecule and DNA is a double-stranded molecule. So only one of the strands of the DNA are coded by the RNA polymerase into a messenger RNA molecule. So only one strand of DNA have to act as, we can call sometimes a template strand. It acts as a template complementary to which a messenger RNA molecule is formed. So we call it a template strand. RNA polymerase moves on the gene and open or unwind the double helix. It do so by breaking down the hydrogen bonds between the two strands. As we know that the two strands of DNA are joined together by the hydrogen bonding. So RNA polymerase break down those hydrogen bonds and open up the helix, the double helix. Now one of the helices act as a template strand. Then RNA polymerase moves on this template strand and adds nucleotides from the surrounding fluid to make a template RNA strand. The difference is this, that only in the place of thymine, a uracil base is placed. All the other bases are same. Adenine, guanine, and the cytosine are same, but in place of thymine, it adds a uracil base. The RNA strand is exactly complementary to the DNA strand. That is, if on the DNA our base sequence is GCA T, then on our RNA it will be against G a C, that is C, against A a T, that is G, in the place of T a U, so G U T T, a different one, exactly complementary to the DNA strand. RNA polymerase, when it moves through the gene, it opens up the double helix. Double helix appear as a bubble inside the double helix of DNA. We call it sometimes a transcription bubble because DNA strands are open in a way that they appear as a bubble. We call it a transcription bubble. In this diagram, you can see that there is a structural gene, a region which have to be coded, the actual gene. Before that, there is a small region which is called a promoter region, which is identified by the RNA polymerase. Behind, you can see the enzyme and just a model, or we can say model or a cartoon diagram. The RNA polymerase, which is attached to a promoter region, but it is extended until the initial part of the structural gene. When it properly attaches to the promoter region, then it starts opening the double helix and a transcription bubble is formed. Then RNA polymerase moves on the DNA strand. It moves on the gene. The helix unwinds more and it makes a complementary strand of RNA. This strand of RNA protrude out of transcription bubble. When messenger RNA start forming and it start elongating, then this comes out of the transcription bubble. We look at a diagram to make it clear. Here you can see a replication bubble, which is actually DNA strand opened by the RNA polymerase, and you can see that in blue color a messenger RNA which is forming is coming out of that transcription bubble. At end of the gene, which means that where the length of DNA ends, which have to code for a protein, there is a sequence of DNA itself which is called a stop sequence, which is actually a signal that now this gene has ended. When the stop sequence arises, usually this is a series of GC, that is GC base pairs, guanosine and cytosine base pairs, followed by a series of AT base pairs. Due to these sequences, these GC base pairs makes actually join together to make a hairpin loop like structure. Just they bind in this way to each other that they make a structure which looks like a hairpin. We call it a hairpin loop. When a hairpin loop arises, then RNA polymerase cannot go ahead, and this is separated from the DNA molecule. This is the termination of the transcription, and the messenger RNA is separated. It goes out of the transcription bubble. RNA polymerase is separated, and the DNA start winding again, just like as it is normally present. So this is end of the transcription. The transcription includes initiation, attachment of the RNA polymerase to the promoter region, then unwinding of the DNA double strand to make a transcription bubble by the RNA polymerase. Then addition of nucleotides, making a complementary strand of RNA by the RNA polymerase by addition of nucleotides. Just there is one difference, that in place of thymine always a uracil will be added. Then release of this messenger RNA molecule from the transcription bubble, and then the end of the sequence, that is the stop sequence, comes. A hairpin loop is formed and RNA polymerase is separated. This is end of the transcription. Now messenger RNA is free to go inside the cytoplasm or is ready for translation. The process of translation is different in prokaryotes and the eukaryotes. In prokaryotes, as we know, that they don't have a true nucleus. They have a nucleoid region though, but true nucleus is not present. So what happened, that whenever their messenger RNA and this actually their nucleoid region, their chromosome is almost continuous with the cytoplasm. So as their messenger RNA comes out of the transcription bubble, it starts translating itself. The ribosomes come and attach to it and they start translating it. But in eukaryotic chromosomes, this is a more complex process because they have a nucleus and the messenger RNA has to travel through the nucleus, move out through the nuclear pores into the cytoplasm where ribosomes are present, and then they have to be translated. So the messenger RNA in eukaryotes is modified. Because it have to move a long path, it is exposed to various enzymes present inside the nucleus and cytoplasm, which are called nucleases and proteases. To protect it from the enzyme action, two modifications occur in the messenger RNA. One is called a cap, addition of a cap, which is a 7-methyl GTP cap. On its 5 end, there is a 7-methyl GTP molecule which is attached, and on its other end, a poly A tail, that is AAAA. Adenine nucleotides are added. These are the two major additions which protect this molecule from the action of nucleases and the proteases. There is another difference between the prokaryotic and eukaryotic DNA. The eukaryotic DNA have some extra sequences inside their integrated in their genes. We call them introns. These introns have to be removed. If we look at a diagram, in the diagram in front of you, you can see that there are in green sequences of DNA called exon, then comes an intron, then comes an exon, and then comes an intron. These exons are actually the parts of genes. These introns are non-coding sequences. These does not have to code. So what happened, that before messenger RNA is translated into the proteins, these introns has to be removed. So there is another difference between the prokaryotic and eukaryotic translation, that before translation, messenger RNA is, we can say, spliced. We call it RNA splicing, breaking down and removal of these introns from the messenger RNA molecule. If down there we have, look, this is a coded gene in which this is a coded messenger RNA molecule in which the introns are removed by different types of molecular processes, events. Next is translation. Translation is from messenger RNA to protein. Messenger RNA have to code itself in the form of proteins. The translation process consists of 3 steps. If we divide it over the length of time, we can call them 3 stages or 3 steps: initiation, elongation, and termination. Initiation is the start of translation. When messenger RNA in eukaryotes comes out of the nucleus through the nuclear pores and enters into the cytoplasm, it encounters the ribosomes. We know that ribosomes are the protein making machinery of the cells. But what happens in prokaryotes, that when their messenger RNA starts forming, coming out of transcription bubbles, it directly comes out into the cytoplasm and ribosomes come and attach to it and start translating it. So in eukaryotes, introns are removed and it comes out of the nucleus, reach the cytoplasm, and then it is ready to be translated. What happened actually? When messenger RNA in eukaryotes reaches in the cytoplasm, the ribosome, which have two subunits, one is small and one is large, the small subunit comes and attaches to a specific site on the messenger RNA. We call it a start codon, from where the actual gene starts. When the first codon is set on the when the small subunit is set on the first codon, then the large ribosomal unit comes and attaches to the smaller unit and the messenger RNA molecule. This larger subunit, as you can see in the diagram, has three sites: E site, the exit site; P site, the peptidal site; and A site, the aminosyl site. E site, from where a transfer RNA have to exit; the P site, where the peptide, where the transfer RNA with the peptide, a whole protein chain which is elongating, is present; and the A site, where the newer transfer RNA comes bringing a nucleotide. Then the first transfer RNA comes and attaches to the start codon. Actually, transfer RNA is an RNA molecule which have an anticodon present on itself. For example, if the start codon is AUG, then on the transfer RNA there will be a complementary sequence. That due to this complementary sequence, transfer RNA bringing the amino acid, because according to the anticodon present on the transfer RNA, it will bring a specific amino acid from the surrounding cytoplasm. Transfer RNA comes and attach to the P site. After that, next transfer RNA brings another amino acid according to the codon read by the ribosome, by the transfer RNA. Next transfer RNA come and attaches to the A site. Then a peptide bond is formed between the first amino acid and the second amino acid. When this peptide bond is formed, then the ribosome moves ahead on the messenger RNA. The result is this, that now the first amino acid is present on the E site and the second amino acid is present on the P site in place of the first amino acid. And the A site, as you can see in the diagram, is free. When this A site becomes free, then the next transfer RNA identifies this site and brings the amino acid according to the anticodon present on this transfer RNA and comes and attaches to the A site. When it attaches to the A site, then again a peptide bond is formed between the second amino acid and the third amino acid. When this bond is formed, then the ribosome again moves ahead. Due to its movement, the first amino acid, the first transfer RNA, which brought the first amino acid according to the AUG codon, the methionine, is released from the E site or the exit site. And now the second transfer RNA comes on the exit site, E site, and the third transfer RNA comes on the P site, and A site is again free. Then again, according to the codon read by the transfer RNA, fourth transfer RNA comes and attaches to the site. Again a peptide bond is formed, and this process goes ahead until a stop codon comes. At the end of the messenger RNA, there is a codon called the stop codon. This codon does not matches to any transfer RNA. In place of the transfer RNA, a protein complex called releasing factor comes and attaches to that site, that codon, and when the releasing factor comes and attached to the codon, the polypeptide is released into the cytoplasm, that is broken down from the transfer RNA. Transfer RNA is also released, and the ribosomal subunits, they are separated, and the messenger RNA is also released from the ribosome. This is how the process of translation completes. So initiation, when small ribosomal subunit comes and attached to the messenger RNA, then the large ribosomal unit comes and attaches to the messenger RNA. Then the transfer RNA comes according to the codons bringing amino acids, and the ribosome moves on the messenger RNA to make a polypeptide chain. At the end, due to releasing factor, they are released. So a new protein is formed in this way. This may be a structural protein or this may be a functional protein. So today we talked about chromosomes, chromatin material, the processes of replication, transcription, and translation. I hope you understand what we talked about today. We will meet in the next lecture. Thank you.